evolution. And we had reached a point in microbial evolution where, I don't know why all these things are now all opening. We had reached a point in microbial evolution where I had already started to talk about the cyanobacteria. This is just an extension of the end of that lecture where we were talking about characteristics of the cyanobacteria, which are eubacteria, which remember are photosynthetic. The classic photosynthetic bacteria in the sense that they carry out photosynthesis in the way in which you envisioned photosynthesis, which is taking CO2, converting indirectly to carbohydrates, and generating oxygen as a byproduct. And if you recall, I also said that the cyanobacteria sit in that middle where we move from an anoxic, or no oxygen, to low oxygen to higher oxygen. So some cyanobacteria actually can carry out anaerobic photosynthesis, which is the type of photosynthesis which is considered bacterial photosynthesis that many eubacteria carry out. And then the cyanobacteria, a large proportion of them, are also key bacteria in terms of cycling, as many bacteria are, and they're very much involved in the movements of nitrates. Hence, they have a lot of organelles, and they have a lot of it's not organelles, sorry, they have a lot of membranes. So they have an extensive membranes called the thylakoids, and basically it's those thylakoids. I'm just gonna. Okay, it doesn't want to turn off the lights at the board, so the thylakoids are where we actually carry out photosynthesis. And then they also have those, so you've seen a picture of this in earlier lectures, they also have this heterocyst. It's amongst usually the chain of cyanobacteria that also have a significant amount of membranes for which they carry out nitrogen fixation. The other interesting thing about the cyanobacteria is many produce a range of toxins. Many of them are harmful, but the interesting thing, some have shown some usefulness in terms of being a pharmacological agent. So it's not that all of the toxins are bad, but rather some of them may actually, as we research these toxins more, might have a very useful role in various types of development of pharmacological drugs. It's an interesting paper if you're interested in it. From a point of view of us in terms of being aware of cyanobacteria in lakes and ponds, remember they are also part of algal blooms. So when you see that kind of green-blue algal bloom that happen on the surface of ponds and lakes, those have the potential. I don't know what's going on.
Yeah, I know. I saw something was wrong with it.
But it's not. It's green. So unless somebody touched something to change it.
That's the problem, right? Yeah.
And I didn't know which button. Because I haven't pushed a button, so I'm wondering.
No, it should be all right. Yeah.
Sometimes this thing gets changed. Was that the right
I'm gonna check that.
setting? Okay.
Well, let's have a look.
Okay.
Anyways, please continue.
Anyway, so it is important to understand that the algal blooms can be potentially dangerous to you. In other words, don't go swimming in an algal bloom. But they're also very dangerous to smaller animals. And many people have allowed their dogs to swim where there have been algal blooms. And unfortunately, in those algal blooms, there were some very harmful toxins, and the dogs died. So you do have to be aware when you're close to bodies of water. If you're seeing algal blooms, it can be problematic, especially if you start to see more of a blue color rather than a green color in that bloom. Did we solve it?
Yeah, somebody had changed the
Setting.
the channel.
And I didn't. Yes, I've had that happen before. Thank you very much.
No problem.
All right.
Sure.
All right. Technology. All right. So from the sense of what I've defined, so remember last week I said that I was going to use the evolution of microorganisms as a way to define different terms. So from last week to what I just covered, I gave you the definition of what prokaryotic was, and what the kinds of characteristics prokaryotes have, because the first organisms were prokaryotic. So now we move to the next characteristic that was true of the first organisms. And that is they were anaerobic. So they did not live in an oxygenated world. It was an anoxic world. And I think a lot of times people think, well, it just means they can grow in the absence of oxygen, but that they're fine if they're in the presence of oxygen. That is not true. The majority of anaerobes will die in the presence of oxygen. So oxygen is actually toxic to them. And it's because if we look at their evolution, they have not evolved the enzymes they need to deal with oxygen as part of a metabolism, which of course we know that oxygen produces what when we metabolize and carry out our functions in our daily challenges? Oxygen-free radicals. And we need enzymes to be able to alter those, otherwise they cause severe damage to cells. So really they grow in the absence of oxygen. There's no oxygen use in metabolism. And we have lots of modern examples of anaerobes, including those that are photosynthetic, as well as those that are actually non-photosynthetic, but they are anaerobic. And remember, again, this is a slightly different take on photosynthesis. Bacterial photosynthesis carried out by all eubacteria except the cyanobacteria is done anaerobically. There's no use of CO2 and there's no production of oxygen. Okay? All right, so it is the second characteristic of the earliest cell types, remember? And what we can have is various forms of utilization of oxygen. So when we say something is an obligate anaerobe, the word obligate means it can only grow under the conditions it's attached to. So if it's an obligate anaerobe, it has to grow in the absence of oxygen. If it's an obligate thermophile, then in fact it has to grow at high temperatures. There are no options in that. So in these cases, the oxygen is often lethal. In fact, under most conditions it is. And many of our human pathogens, including the pathogens that we find within Clostridium, which is a gram-positive rod that forms endospores. And two of the very important members of that genus is Clostridium botulinum and Clostridium tetani. So botulinum causes botulism, which often happens in inappropriately canned or stored foods where you have spores forming and then you ingest the spores, so the spores are present. You ingest the spores, which in theory are not lethal to you until they actually germinate in your body to form a vegetative cell, and then they make you very sick. Okay, the same goes with tetani. Tetani is the cause of tetanus, and again, tends to be more problematic in deep wounds. And keep in mind that because it's a spore former, but it's anaerobic, it's less of a problem in terms of being a bioterrorism threat. But the bacilli, or the Bacillus genus, for which you've already met in the lab, they form spores too, and many of the Bacillus species actually have potential to be very harmful. And so you can send spores to somebody, they inhale the spores germinate in their lungs, and they end up with a severe infection. So in fact, endospore-forming bacteria have been used in bioterrorism attacks. One of the major ones was actually in the United States when people sent them to various politicians who then inhaled, but they stopped that plot before anybody was actually made sick. All right. But we also have a group of organisms that are called facultative anaerobes. So when we put the word facultative in something, it means it can be an anaerobe, but facultative anaerobes prefer to grow in the presence of oxygen. So they're facultatively capable of it, but they don't grow optimally. So they grow better in the presence of oxygen because obviously, as we know, more and we remember our cycles, more ATP is produced in the presence of oxygen. And a classic example of a facultative anaerobe is E. coli. Prefers to grow in oxygen, grows fast in the presence of oxygen, but if you reduce its oxygen or take it out of oxygen, it still can grow. And actually, this has relevance when you start doing your experiment where you look at the generation time of E. coli, which you will start in lab week 5, where you'll compare the growth of E. coli at 2 different temperatures. And you're going to think about how you're growing and what you need to grow. So, for example, if you're sampling from a flask to look at growth, and you're taking the flask in and out of a water bath, and you're forgetting to agitate the flask on a regular basis, and you're also taking it out of the water bath, do you think the flask is at the right temperature or the right aeration? No, because shaking of a culture creates aeration in the culture. So if you have a stagnant culture, actually you're not generating air in that culture. So keep that in mind when you're doing that experiment. There are eukaryotes that fall under this as well, and Saccharomyces cerevisiae is actually one of them. And they undergo, under anaerobic conditions, they actually carry out fermentation. And that fermentation is what gives you the alcohol that is so useful in various products that Saccharomyces cerevisiae is involved in. All right. And then we have a couple of other categories. The next largest group in terms of oxygen requirement is called the aerotolerant. And aerotolerant means it tolerates oxygen, but it doesn't need oxygen. And so it grows equally well in the presence or absence of oxygen. A classic example is Streptococcus pyogenes, which is the bacteria that's responsible for strep throat, as well as the flesh-eating disease that we'll talk about when we start talking about the gram-positives and the issues around major gram-positive pathogens. All right. Now, there's one other group, which I'll mention here and then come back to. There's also a group of organisms that are called microaerophilic. So can you guess what maybe microaerophilic means? Micro in front of aerophilic. So aerophilic would mean oxygen, so microaerophilic might mean they need oxygen, but at a much lower level than is found in the atmosphere. So you might predict then those organisms may have evolved between the transition between an anaerobic world to when the world started to become oxygenated, when there were low levels of oxygen present. And they still exist today. So when we talk about the concept of these organisms versus an aerobe, remember, an aerobe utilizes and requires oxygen, and some will actually not only require the presence, remember they require the presence for growth, although some aerobes, which is where I'm going to reiterate something, are actually what we define as microaerophilic. So they require oxygen, but only to about 2 to 10%, which is lower than atmospheric oxygen, which is usually around 20%. Classic examples of these are Treponema pallidum, which is the causative agent of the sexually transmitted disease syphilis, or Helicobacter pylori, which can overgrow in your stomach. So it has the ability to grow in the presence of acid, but it overgrows when your stomach becomes too alkaline. Okay? And your stomach is not renowned to being highly aerobic. So it grows in the lower presence of the oxygen, originally tied to gastric and duodenal ulcers. So originally, the idea of ulcers, if you go back in time, was they just form because you're under stress. No, ulcers don't just start because you're under stress. They're actually caused by Helicobacter pylori. And this bacteria has also been tied to an increased incidence in some individuals of stomach cancer. Okay? So they are present in everyone's gut to a tiny bit, but they really are not problematic for most people, but they again can undergo overgrowth in certain individuals based on the conditions of their stomach. And this was one bacteria we knew nothing about for the longest time, because if it's a microaerophilic organism, it's very hard to actually hit the right conditions under which they will grow. So anaerobes and microaerophiles are well, if we think about the world becoming oxygenated, would have evolved before the aerobes, when the Earth was just beginning to change from an anoxic or anaerobic environment to an aerobic environment. And we can actually do an experiment in a lab to demonstrate these different oxygen requirements that bacteria have. So every organism you're using in the lab is an aerobe. And the evidence of that is because you're putting it in a tube, you're putting it in an incubator, and it's either being shaken or left alone. You're not creating conditions where you're removing oxygen. So we're only dealing with aerobes in the lab because we have to use much more sophisticated growth. We have to use anaerobic jars. We have to create incubators that can control the levels of oxygen, and there's no point for us to do that for the purposes of understanding microorganisms. But we can do an experiment that will help us identify these characteristics. And this is done with a very specific media. And this media, which will allow us to actually identify whether an organism is an aerobe or an anaerobe, is basically it's similar to the motility tube and the gelatinase tube that you will use, where when it solidifies, it doesn't actually solidify solid, so it allows bacteria to move, so it's a semi-solid media. So if you actually look at the motility tube, it actually isn't put at 37 degrees, correct? You left it out in room temperature. Why? Because if you provide a little bit of heat, the media will start to melt, because it's designed to allow the bacteria. Because what did you do in your motility tube? Where did you inoculate in the motility tube? Yes, inside that tube. And you didn't go all the way to the bottom. You went to about 3/4. And if it's a non-motile bacteria, it will only stay there. But if it's motile, it can move out of that tube, and you'll get red colors throughout the tube because it goes to the bottom and comes out that tube because there's a hole in the bottom, and it can move to the rest of the media. And so if you see red outside of the tube, importantly, that starts at the base of the tube and goes this way, not if it's just as you did a poor job of inoculating, you flipped a bunch of bacteria onto the surface of the tube, then you have a motile bacteria. That's not possible to do if you use the same agar that you have on a nutrient agar slant, for example, because the bacteria can't move on that solid surface. So this media is semi-solid, and what you do is you create this, take this media that also has the ability to basically eliminate oxygen, and you end up with an oxygen gradient, and you end up with a region that's pink at the top because there's an indicator that says there's oxygen, and then there's a reducing level. And what you do is you take the media before it solidifies, you add the bacteria, you mix them all together. So in essence, you're inoculating the bacteria throughout the tube. Then you put it in the incubator and you ask, where does the bacteria grow? So if it's an aerobe, it's only going to grow at the top of the tube because there's only oxygen at the top of the tube. If it's an anaerobe, obligate anaerobe, it's only going to grow at the bottom of the tube. If it's a facultative anaerobe, it likes oxygen, so it's going to grow more at the top, but it will be possible to grow throughout the tube. If it's in fact a microaerophilic organism that likes slightly lower concentrations of oxygen, it's not going to be right in the pink area, but it's just going to be slightly lower where the oxygen levels are lower. And then if it's an aerotolerant organism, it's going to grow throughout the tube because it doesn't care whether oxygen is present. So you can do this. It's called thioglycolate media. We're not going to do it in lab, but you can do it. It's a semi-solid media that then will solidify after you've inoculated. You put it at room temperature. You watch where the organisms grow. So you can demonstrate this oxygen requirement. And really what it comes down to is, do these organisms have the two important enzymes that allow an organism to live in the presence of oxygen? Superoxide dismutase and catalase. So superoxide dismutase, remember, takes the oxygen-free radicals, produces oxygen and hydrogen peroxide, and then that catalase takes hydrogen peroxide to deliver water and oxygen. So the organisms would have had to evolve these enzymes. And remember, over the course of time, the ability to do different things is based on the ability to evolve the enzymes that can carry out that function. So these are actually called agar shakes. So really, what you end up with is, for example, if I had done this experiment with streptococci, which are aerotolerant, by the way, or with enterococcus, which is also an aerotolerant bacteria. Enterococcus is, as I've already pointed out to you, naturally is present in your intestines, where again oxygen levels often happen to be lower. Then I would have seen this kind of growth. So really, what it comes down to is the two enzymes that we often talk about is in physiology and metabolism as being critical for us to survive in an oxygenated world. Because otherwise, the oxygen-free radicals that build up during metabolism cause enormous damage to your cells. Hence why you're encouraged to take certain vitamins that would help you deal with oxygen-free radicals, okay? All right, so again, these two enzymes are necessary for this. If they don't have them, then in fact, they become toxic and the organisms will not survive. So aerobes are going to have both of these. Many of the facultative anaerobes, like E. coli, actually have catalase but lack superoxide dismutase. So they have some of the enzymes, or some of them have superoxide dismutase and they lack catalase. And in a subsequent lab, you're going to actually do a catalase test on a gram-positive and a gram-negative and see the different levels of catalase, which will pair, by the way, to a disinfectant lab, of which one of the disinfectants you use to show killing effect is hydrogen peroxide. So hydrogen peroxide is a common antiseptic, as well as a disinfectant at higher levels, that we use to kill bacteria. So the question is, does it kill all bacteria? So you have to think about this. If, in fact, the bacteria produces a large amount of catalase, will hydrogen peroxide be as useful a tool to kill that bacteria? And the answer will be not at low levels, because the bacteria has enzymes that can destroy the catalase. So that will come up after reading week, where you do that experiment. The third characteristic of the earliest cell type was, if you remember, being heterotrophic, which means they have to consume their organic material. They weren't photosynthetic, so they weren't autotrophic, they were heterotrophic. So heterotrophic simply means, heter means different, trophic means nourishment. So they require carbon in some organic form. That's very important. In an organic form, they can't use carbon in an inorganic form. And of course, the most useful form of that carbon for all organisms, the easiest sugar to break down is glucose. Okay. And of course, they can use other organic carbon molecules as well. So heterotrophs cannot use CO2 as a major source of major carbon source to make their nucleotides, amino acids, and fatty acids. But remember, a misconception of photosynthesis is CO2 is directly converted to the carbohydrates, but it is not. It goes into the Calvin cycle. You make that 3-carbon sugar, then it goes and actually is added into the pathways where you end up making the actual sugars. Actually, photosynthesis to make the first carbon molecule is actually an energy requiring process. It is not an energy making process. So actually, the first steps of photosynthesis actually tax the plant, because they actually use up energy to actually make the first building block to actually make their carbohydrates. All right, so all organisms are heterotrophs except for autotrophs, which of course can use CO2 and indirectly produce your carbohydrates. But within this also, there is a couple of different types of autotrophs, but there's also a couple of different types of heterotrophs. So again, I'm defining terms that you have seen in high school. You've seen it in first year biology, and now I'm just reminding you of these terms. So you also would have seen it in some of your second year courses. So a photoheterotroph can use sunlight as an energy source, but has to have an organic source of carbon. An example is the purple non-sulfur bacteria and the green non-sulfur bacteria. Within the bacteria, there's also the chemoheterotrophs, like we are. We are chemoheterotrophs because we require organic compounds as an energy source as well as a carbon source. So there's a small subpopulation of heterotrophs that actually can use sunlight as an energy source. Now, again, and hopefully you all remember this, and this is just a review, the autotrophs are using CO2 as a major but not direct carbon source for the biosynthesis of cell constituents. CO2 is first fixed into a carbohydrate, which then becomes the source of the carbon skeletons. So photoautotrophs use sunlight as energy. So that would include the cyanobacteria, the purple sulfur bacteria, the green sulfur bacteria. These bacteria, the purple sulfur and the green sulfur, are anaerobic bacteria. They carry out anaerobic photosynthesis. Okay? The chemoautotrophs or chemolithotrophs, the cyanobacteria, to extend that, by the way, can use sunlight as an energy source in an oxygenated world. But these purple sulfur bacteria and green sulfur bacteria are actually carrying out anaerobic photosynthesis. The chemoautotrophs are sometimes called the chemolithotrophs, which are not talked about in great amount, and I'm not going to either. They're a very small group of very important bacteria because they use inorganic compounds as an energy source. Okay? So there is a variety of different types of heterotrophs and autotrophs amongst the bacterial world. And we have lots of modern day examples of autotrophs. And by definition can use carbon dioxide as a carbon source and usually light as an energy source. Green plants, eukaryotic algae, cyanobacteria. The photosynthetic bacteria are different from the cyanobacteria because they do not use carbon dioxide as their carbon source. They actually use H2 and H2S as electron donors versus what we see of using H2O that is used by cyanobacteria, which I'll show you in a minute in a diagram. Remember, certain types of non-photosynthetic autotrophic bacteria are called chemolithotrophs. Many of those are going to be autotrophs. Basically, except for the autotrophs that I listed here, all the other organisms in the planet really are heterotrophic. So we rely on the autotrophs actually substantially to provide the building blocks for the heterotrophs. So the chemoheterotrophs or the organotrophs are the most common group. We fall within this group. We use organic compounds for energy source. We use organic compounds as a source of carbon. So we are relying on these small microorganisms and to produce a lot of building blocks that we actually need that can carry out different types of metabolism. So just to complete the chemolithotrophs without going into a lot of detail, in some books they're called the rock eaters, chemical rock eaters. So they do use carbon dioxide as a carbon source, so you can call them a chemoautotroph. They're only found in the prokaryotes. They're widely distributed amongst the eubacteria and the archaebacteria. They get their energy from inorganic compounds, and each of the cell types usually specialize in different inorganic materials, from hydrogen gas to H2S to various forms of iron or ammonia. They can also use, and they're thought to have, and this is just a short list of the various materials they can use. And they really have their evolution is thought to have occurred from cyanobacteria. So we went from a aerobic photosynthetic organism to one that can utilize a chemical form of autotrophy. So they didn't evolve it early. They actually evolved after the cyanobacteria. And where they live is in very, obviously based on what they use, very inhospitable environments, such as sulfur hot springs, which are rich in reduced inorganic compounds, such as hydrogen sulfide. And we have a lot of modern examples of these, mostly, well, there are eubacteria. Many of them are in the archaebacteria, where they can derive energy from the metabolism of organic substance, but also can derive energy from inorganic substances. So we have examples in modern day of organisms that can do both processes, depending on where they're actually living. All right. So just as some examples, and again, these are just a couple of examples. We have sulfur oxidizing bacteria that can oxidize metal sulfates, or H2S, to SO4 2 minus ions, or nitrogen oxidizing bacteria. So this is different from the nitrogen reducing bacteria, where they can oxidize NH3 to NH2 minus, which are nitrates, or to nitrates. These become the nitrifying bacteria. We have iron oxidizing bacteria, can oxidize both forms of the iron. And again, they tend to live in extremes environments, so many of them, not all of them, but many of them, the majority of them even, are archaebacteria. And so you could imagine then again, and if you go back to the cycles and look at the names and look at the characteristics of those organisms, and I'm never going to ask you that question, so don't go look at the cycles and say, oh my God, I have to know all the names there. No, you do not. But if you were to do that and go back and look, what you'll see is these bacteria that fall under this group are critical in biogeochemical cycling. So again, the role of microorganisms is very broad and very significant to creating the environment in which organisms grow, and creating environments in which plants can grow, as well as other organisms. So if we actually go back to what I talked about last week and say, well, what is the evidence that the first organisms were prokaryotic, anaerobic, and heterotrophic? Well, really, it's based on deduction and some evidence. It doesn't make sense that the first cells were more complex, and then simpler cells evolved from complex cells. So the first cells were likely prokaryote because of the size and simplicity of those cells. And there's other deductive reasoning we can use to try and understand that. Since eukaryotic cell structure is relatively complex, prokaryotic cell structure is relatively simple. You notice you never hear me say prokaryotes are simple organisms. They are not. They are still very complex. They are just not as complex as eukaryotic organisms. So simple most likely evolved before complex. And there's very good evidence, and I've already given you some of this. As the first cell-like systems evolved, they would have evolved in an environment where the oxygen was not freely available, so that would suggest the first organisms were likely anaerobic. And then, when we ask the third question, which is, cells of most organisms contain several identical metabolic pathways. So all those pathways that you learned are in almost all organisms. In fact, they are in all organisms. But some of them that are aerobic respiration are not done by anaerobic organisms. Okay, so the key point is we do know that one of the cycles can function anaerobically. And what is that cycle? Sorry? Glycolysis. So therefore, these organisms could have survived because there are metabolic pathways that can function in the absence of oxygen. Additionally, the use of an organic form of carbon as a source of carbon skeletons for synthesis, where we would have had to have various materials made, required us to generate pathways. So biochemical pathways evolved in a stepwise process. And the first pathway that would have evolved would have been glycolysis. And when you look at glycolysis, you see that. It's identical in all organisms. It's a 9-step process that can be an anaerobic pathway, which also supports this idea of being anaerobic and heterotrophic. But the point about the metabolic pathways is also very important, because in order for us to develop in order over time for these various abilities of organisms to evolve, different biochemical pathways had to evolve, and different enzymes had to evolve over time, because the first cell types did not have those. And remember, this would have been taking place in all sorts of locations. So we started with that, what was defined as that primordial rich soup in terms of being organic, but the synthesis of those organic molecules was done as a chemical reaction, not as a biological reaction, but could accumulate over many years because there were no users. Those heterotrophs then, once there was the first step of evolving the first cell types, could evolve and rapidly utilize the nutrients made by abiogenic processes. But a problem is going to arise, which I've already talked to you about. Eventually, there would be too many users. And the chemical reactions are too slow to generate enough material for those organisms to be able to survive. So then this would require certain organisms to evolve pathways. So once that rate of nutrient utilization was much greater than the rate of abiogenic synthesis, certain essential nutrients became limiting, which would have provided selective pressure. And over time, certain organisms would have evolved through evolution certain abilities. So cells with pathways able to synthesize a missing essential nutrient from an existing nutrient world would have a selective advantage, but it would have had to been an ordered process of survival. Okay. So for example, if nutrient Z, which is the end of the pathway, okay, so I'm taking you through a pathway and realizing pathways would have evolved from the last step to the first step. So nutrient Z was plentiful in the primordial soup. When heterotrophic systems evolved, nutrients evolved, it would have been used up, and nutrient Z would become essential. It would be used up very quickly, therefore it became depleted. So therefore, organisms that evolved the ability to make Z would be more successful. Okay? They would be able to survive. But then after a period of time, once Z was depleted, cells could use the Y precursor if they could make Z, right? But at some point in time, Y would become limiting. And only the organisms that actually could make Y would survive. Okay? So therefore, those organisms that could make Y and Z themselves with enzymes using precursors would be successful, and those cells would predominate. This is deductive reasoning in terms of developing pathways. If Y was depleted, then again, the cells would not be able to survive because they couldn't use Y to make Z, and they needed Z because it was essential. Then any organism that could make the next earlier step in that pathway, which would be X, would be successful. And what are we building? We're actually building a metabolic pathway. And so with each one of these steps, when X is depleted, then you have Y, and it would develop Y. In the end, you evolve biosynthetic pathways that allow you to start with simpler and simpler and simpler building blocks. And so really what you have is over the course of time, the evolution of biosynthetic pathways that allowed organisms to be able to survive and make biologically the materials that they needed. All right. So again, the biosynthesis of organic substances from CO2 by autotrophs would be much more rapid than their synthesis by abiogenic reactions, would restore organic substances to the biosphere. Organic substances would be available for use by heterotrophs, permitting the coexistence of heterotrophs and autotrophs. So the autotroph evolution helped the survival of the heterotrophs in terms of developing those pathways. So heterotrophs actually depend on autotrophs for survival. How could aerobes evolve from anaerobes? That's much more much more readily, perhaps, understandable. You started with an anoxic world, so the first autotrophic pathways were likely anaerobic. You have examples of those that still exist. They carried out bacterial photosynthesis. This is not what's carried out by the cyanobacteria, but they do, but they still use chlorophylls, and it's an anaerobic process. It involves the use of reduced sulfur compounds to ultimately fix CO2. So the anaerobic photosynthetic bacteria also have a Calvin cycle, because it's the Calvin cycle that actually generates the carbon. Okay? But they have a different system of generating the processes to go into that Calvin cycle. So they use H2 from H2S to reduce CO2 into carbohydrates. In the process, they release free elemental sulfur or other sulfur compounds. So this becomes the reaction of making carbohydrates, the general reaction. So what's the difference between that and what you've learned about aerobic photosynthesis? Instead of H2S, what's in that simple formula? Water. Okay? And what's this? Not elemental sulfur, it is oxygen. All right, so really it became a change in what entered the system and what was the byproduct of that system. So aerobic photosynthesis uses chlorophylls and uses H2O to reduce or fix CO2. Cyanobacteria, which are all aerobic photosynthetic autotrophs, are presumed to have evolved from anaerobic photosynthetic bacteria, which used H2S to reduce fixed CO2. So using that led to the formation of carbohydrates. So the cyanobacteria that evolved early would have also been using this pathway until they subsequently evolved the ability to generate, the cyanobacteria would have evolved out of the anaerobic photosynthetic bacteria to actually carry out this pathway, and then, of course, subsequently evolved into eukaryotic algae in higher plants using the endosymbiont hypothesis, which says that there was an endosymbiotic event where you had phagocytosis of cyanobacteria into a eukaryotic cell that led to a cell that could carry out photosynthesis. So when we think about the evolution of cyanobacteria and go back to some of the tables, we need to remember that they first evolved when there was little or no free oxygen, so that's why some of them have the ability to carry out anaerobic photosynthesis even today. Again, we know that what we know about oxygen, yes, it creates oxygen-free radicals, but what else do we know about the generation of oxygen and what it would have done to the atmosphere? It's extremely reactive, and it oxidizes in the presence of UV light to give ozone. So what would have evolved as the world became oxygenated? The ozone layer that wouldn't have existed before. That has relevance because if the ozone, what does the ozone layer actually do? It prevents UV. So in the early evolution, there was no ozone, so you had UV light causing lots of mutations over very quickly, causing a very quick evolution of different organisms due to mutations. Remember, it wasn't necessarily a planned mutation, but a mutation occurred, and that organism had an advantage. And so that organism ended up being in a higher number than another organism. It wasn't that the organism figured out how to mutate its cells, but rather mutation was occurring as it happens randomly, and it led to some evolution of cells that could carry out certain functions. So as the free oxygen levels increased due to cyanobacteria photosynthesis, really we ended up with the ozone layer. The reducing atmosphere of the planet changed to an oxidizing atmosphere we have today. So if we look at cyanobacteria and the pathway the pathway they use, here it is here, and this photosystem electron transport chain feeds the material into the Calvin cycle, which leads to the formation of the 6,3-P, which ultimately gets converted to various nutrients, including starch, and can also be used for amino acids and fatty acids, and ultimately you have the export the sugar. In the terms of the anaerobic bacteria, instead of using H2O, they used H2S, and instead of using oxygen, producing oxygen, they would have produced elemental sulfur. So really the difference is what was used to start the photosystem and what was actually generated as a byproduct of that system. So you can start to see that a change here in terms of the ability, a mutation that would suddenly allow some organisms, the ability to use H2O, would have started the process to produce those organisms that were capable of producing oxygen as a byproduct, and of course be able to carry out typical photosynthesis. This all leads us to how do we put this all together? Sorry, I have to change the battery. You all think this is easy, but when you record a lecture, you have to pay attention to what the battery's doing and what the computer's doing and all sorts of fun things. And then you tell me when you do oral presentations, this is hard to talk for 10 minutes. All right. So just to end this part of the lecture, I'm just going to end with an understanding of what, in very simple terms, the endosymbiont hypothesis proposes and what we mean by an endosymbiont. So an endosymbiont is a symbiotic association that's favorable between different cell types in which one or more of those endosymbionts ultimately form a stable association. So we can have symbionts where, so for example, symbionts that may have a mutual relationship and a positive relationship. They may even enter the other cell type briefly, but they can go back out and be independent cells. In the case of the endosymbiont hypothesis, eventually those that form that relationship cannot leave again because they are no longer functional cells. So the organisms that generated the mitochondria are no longer functional cells. Those that created the evolution of the cyanobacteria are no longer independent cells. So there are many examples of endosymbionts that form relationships but do never become part of the organism and lose genetic material. And one good example is the rhizobium bacteria and their interaction with the roots of plants. So they actually are endosymbionts. They go into the root hairs. The root hairs provide them protection and the environment that is low in oxygen so they can carry out the nitrogen fixation to support the plant. But the rhizobium can leave again. It's a free-living cell. It can leave the plant cell. But in the evolution of the mitochondria and the chloroplast, eventually the organisms that formed that liaison lost genetic material, and they no longer could leave, and they became organelles as opposed to free-living cells over many, 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 many generations. So there are several variations of the endosymbiont hypothesis, and I'm not going to get into, a discussion of the various theories of different evolutionary biologists. We're just going to concentrate on that which is fairly well established that ties it back to the bacterial world. And we do see modern day examples, by the way, of the first cell type, which was a large heterotrophic cell that had a nucleus but did not really have any real organelles. And the example of this organism, by the way, in present day is Giardia. It's a eukaryote that has a nucleus but no mitochondria. It's a parasite that grows inside cells, other cells that causes Giardia, giardiosis, which actually is a major cause of gastrointestinal diseases. And if you drink much water, even in Ontario, out of ponds and streams, you very much can end up with a very upset stomach because of the presence of this unicellular organism growing inside of other organisms. Now, this doesn't demonstrate to us that these have existed since the start of evolution. What they are examples of are organisms that lose organelles because they form relationships with other organisms, and they don't need those organelles anymore. So it's demonstrating that the organism could have existed, but that in fact it evolved later in life, the evolution of organisms. So let's just do this, very simply. So the first organism, the first ancestral organism was anaerobic, and it had a nucleus. And as I've already pointed out to you, likely the first organism that was heterotrophic was an archaebacteria. And that nucleus, and they were the evolution of the first nucleus. And a double membrane was formed, and then once this took place, at a later point in time, there was actually an engulfing of an aerobic bacterium. So this was after the oxygenation of the world, or the planet, I should say. And in fact, then we ended up with a phagocytic event that led to the taking up of an aerobic bacterium that over time became which organelle? The mitochondria. And then we had an aerobic eukaryote. So we have a symbiotic system that can now carry out aerobic catabolism. And over time, what would have happened is some of the genes from this aerobic bacterium would have moved to the nucleus. Remember, the eukaryotic cell is diploid, generally. There are exceptions to that rule. And over time, some of the genetic material in the mitochondria, the aerobic bacteria that became the mitochondria would have been lost. Some of the duplications would have been lost. Other genes would have moved to the nucleus. So does the mitochondria have all the genes it needs to make the enzymes and proteins it needs inside the mitochondria? Is the mitochondria self-sustaining? No, it is not. Most of the genes are actually encoded in the nucleus. But there are mitochondrial encoded genes that are made in the mitochondria off the ribosomes that the mitochondria have. And those mitochondria have the same size and characteristic of bacteria ribosomes. Okay? So when we say antibiotics don't affect our cells, they don't have a major effect, but they can affect the protein synthesis of mitochondria because they have the same type of protein synthesis as prokaryotes do. All right, so now we have an aerobic eukaryote, so we have a non-photosynthetic eukaryote at this point. At some point, there would have been a second endosymbiotic step in the primary endosymbiosis where a photosynthetic cyanobacterium was engulfed, and again, the same process. For many generations, it would have been able to go in and out, and our evidence suggests this because we can see multiple membranes around many chloroplasts that indicate there was a constant movement of multiple cyanobacteria in and out. But ultimately, we ended up with a photosynthetic eukaryotic cell, and again, that was now a plastid or a chloroplast, and again, it maintained some of its genetic material, but it transferred some of its genetic material to the nucleus, and it lost anything that was a duplication. Okay? So in the end, what we have is that we have now an organism that can obviously utilize oxygen to generate much more ATP. And the important thing to understand is there is genetic material that is being transferred to the nuclear lineage from both the mitochondrial lineage and the chloroplast lineage. And that's why some of their genes that they absolutely need for functioning are made in the nucleus, and some of them are still made in the mitochondria or the chloroplast. Can mitochondria divide all by themselves? Independent of the nucleus? Think carefully. What do you think the answer is? They have genetic material. Can they divide? Yes. During different parts of your metabolism and your nutrients available, your cells make more mitochondria because they need more mitochondria. And other times there are less mitochondria in the cell. Can chloroplasts divide in a plant cell? So does every plant cell have the same number of chloroplasts? No. Chloroplasts can divide independently of the nucleus. So they are able to divide within that cell. They're able to make proteins, but they also are dependent on the nucleus to produce the mRNA that then will make some of the proteins that they need to be able to function. So those characteristics tell us, again, if we look at the ribosomes of a chloroplast, they are the size and characteristic of prokaryotic ribosomes. And if we were to test them against antibiotics like tetracycline, chloramphenicol, streptomycin that affect protein synthesis, the protein synthesis in the mitochondria and the chloroplast would not function. It would have no effect on the synthesis of proteins that which were encoded by the nucleus. So that information tells us, yes, they did evolve from that. Those two organelles evolved from two different types of the eubacteria. There are two hypotheses, and I really don't want to get into the weeds of this because this is not an evolutionary course, and it still is it's made it to a textbook, which means most people will accept there may be two models. So in the first model, these are competing. The Archaebacteria was the predecessor to the eukaryotic cells. It forms the nucleus. It starts to form some of the important players in the cytoplasm. And then the alpha protobacteria, which are the aerobic bacteria, end up being engulfed, presumably probably as food, but then evolved into the mitochondria. Okay, so you ended up with a cell that had a nucleus and a mitochondria. And really, over time, then you would have developed all the relevant needs. More of the cytoplasm would evolve. The mitochondria would be capable of carrying out its synthesis of some proteins, but some of the DNA would move to the nucleus. So it was no longer a self-contained cell. This, by the way, does not address the chloroplasts at all. The alternative hypothesis is a hydrogen hypothesis, and that's because of some of the very rudimentary mitochondria have some functionality in terms of the use of hydrogen. So again, we see an Archaebacteria in blue, but we also see very quickly a cell that can carry out hydrogen functionality in terms of it can utilize and produce hydrogen. And these two bacterium became associated based on the ability of utilizing hydrogen and CO2 as fuel, and really from this methane-producing bacteria. So the Archaebacteria in general were always generally thought to be the methane-producing Archaebacteria that evolved. Again, because when did this start to evolve in environments that were less than pleasant environments? These would have eventually formed an endosymbiotic relationship where, again, the same thing took place. This bacterium became an organelle. DNA was lost, but had the ability to carry out various functions, and you end up in the same place. From a purpose of understanding the endosymbiotic hypothesis, really what it comes down to is the archae's genetic material likely was the predecessor to the nucleus, and you needed two different cell types to come together to produce the mitochondria. And whether it's this pathway or this pathway remains to be completely established, because you still have different evolutionary biologists suggesting different things. And you will, by the way, still have arguments made by other evolutionary biologists that the nucleus was not the result of the Archaebacteria, but rather viruses or other scenarios. But it makes no sense that it was a eubacterial DNA that was the predecessor. Because as I've already told you, the Archaebacteria have characteristics, or some of them do, of the eukaryotic DNA. They have more than one origin of replication. They have more than one type of RNA polymerase, although those clearly have evolved more specialized functions in eukaryotes. All right. So really, now we're going to turn our attention to viruses after the break. So where can we find the viruses? We never see it on the tree of life, and that's because they're really not living, right? They require a host to survive. They can't survive outside. But we know that the first cells were, at this point, hypothesized to be RNA. The universal ancestor is still DNA-based, and we see the evolution of the bacteria, eubacteria, and we see the line that has the archaebacteria and the eukarya, which tells us that the archaebacteria and the eukarya are closer related than the eubacteria. But the question still evolved, question still asked, where did viruses evolve? And likely, some form of viruses evolved from the first cell types. Because RNA viruses can be RNA-based, they can be DNA-based, they can be double-stranded, they can be single-stranded, they can be linear, they can be circular. So likely they evolved early as the cells that could support them actually evolved. All right. So we note that at the branch point, at this point, regardless of whether there was a direct split or whether the archaebacteria came off the eubacteria lineage, because you'll still see some trees that do that, the first organisms would have been, at the branch point, hypothermophiles, because the world was very, very, very hot at that point. And amongst the eukaryotes, which I already showed you, down here in the diplomonads is that they would have evolved, but they have likely, the diplomonads have lost their mitochondria. So they're at the root of the eukaryotes, but it's not because they evolved without a mitochondria. Likely, they lost the mitochondria during the process of evolution. How do we actually do the genetics to figure out which organisms are related? Okay, so the batteries that I put in are dead. All right, how do we know that there's relatedness in organisms? Well, we use the process of sequencing the ribosomal genes to do that. I don't want to go into vast detail because it's a process where we isolate the DNA, we carry out PCR, we amplify what is the small RNAs that we find in the small subunit of the ribosomal RNA, which is 16S or 15S, depending on where you're reading, which is prokaryotic base. And you look at their sequence, and you discover whether or not they're related to each other or not. And that's how we actually do the genome typing. So generally, all genome typing is done with ribosomal RNAs, DNA that encodes the ribosomal RNAs, and then they often will move to, using other genes, including housekeeping genes, to make comparisons. All right. At this point, we're going to change topics. We're going to start talking about viruses. This is not a virology course. Some of you probably took virology in the fall. I'm going to concentrate on very general characteristics that relate to trying to understand where the viruses fit, and we're especially going to concentrate on the bacteriophage, which you will do an experiment with in week 4. So after you finish your unknowns next week, you'll actually do an experiment with bacteriophage. So it's 1/4 by that clock, so come back at about 25 after. you're interpreting that it's something has changed and the cell needs more mitochondria, then yes, there's a signal. But that doesn't necessarily mean it's necessarily coming out.
But like the mitochondria, they just like survive like on their own. Like just without the nucleus anymore. I guess it's that's like the
Yeah, but a nucleus divides when?
very much like
And what happens?
the cells
So that's not the same as making 5 mitochondria one time, and then because it's under stress, it makes 10 mitochondria.
but then if mitochondria just do that without the cell split, like there'd be like a whole billion mitochondria just like
No, because there still are environmental signals that resulted in a signal that results in it saying, due to the stress the cell is under, the more mitochondria are needed. So very often there's cancer that's producing mitochondria. Okay? You have to put all the organisms you separate. So you start there, so then you do this, and this one's going to yes, but you have to put wherever's not there. Yes, so you have one grandpa, one grandpa, sorry. You have bacillus, but it's not isolated because you also have another grandpa. So bacillus and staphylococcus and micrococcus and micrococcus. It's not until you do morphology, morphology coming off of red stream separating the collites and rods. Because the more the bacillus is positive, you've decided that you decided it's a rod. You don't know that yet. All you got is a red stream positive. There's a morphology step. Where are the rods? Where are the collites? Then all the organisms need to go really you draw the line. Here he says he all of those. So yes, you're supposed to use the least amount of steps, but you still have this is an incorrect statement. You understand what I'm saying? You would have to say positive and wrong. So you would have to say I've gotten rid of all the compounds. So where are they on the switch? How do they fix it? You show where things are. Okay? Western blot is when you want to look at Western blots are really looking at antigens, which are proteins, not in a gel, and you use an antibody. So if you want to determine is a protein present or is that protein accumulated, you run the proteins and then you try, and then you use the antibody to detect that protein and look at its levels. Yes. That's just a good answer. You're welcome. Well, that just tells you that you're not that you have to make sure the stain covers everything. 95% covers everything. So when you're decolorizing, for example, a lot of students don't make sure that 95% covers. So you shouldn't, when you're flooding with 95, you shouldn't be holding the slide. You should have the slide on the left, and it should go on there, and it should flow off the side. I wouldn't say all you do is try to find some space. My guess is it was there. You just Okay. We'll get started and we'll start talking about viruses. All right. Okay, so we're going to sort of go through the general descriptions of viruses, and I'll talk about some viruses that are interesting, that you may have heard about, not necessarily that you may not have actually covered during C20. All right. So remember, cellular microorganisms. Cellular microorganisms, we have been describing the eubacteria, the archaebacteria. They include the prokaryotes, they include the eukaryotes. But we do have a lot of what we might call non-cellular or acellular agents. The viruses, viroids, which are variations smaller than the viruses, prions, which are protein-based. In the world of bacteria, plasmids and transposons can move from cell to cell. So by definition, they are acellular agents carrying characteristics that can alter the host that they enter, which is the definition of an infection. Okay? All of the non-cellular agents are infectious. They can move. Although in some cases, we don't necessarily always understand their full mechanisms of transmission. So for the plasmids and the transposons, we keep seeing variations that these cells are capable of using. It's not cells, sorry, that these acellular agents can use to be able to move. Again, I always give the students these various places you can find information, whether it's the outbreaks about COVID-19 or the outbreaks about measles, which, by the way, there's more outbreaks of measles. And right now, there's a very big outbreak of what virus? Anybody read the news? That makes you have a very upset stomach and a gastrointestinal. And if you live in Guelph, Guelph University in residence, you're not a happy camper. The norovirus, yes. So this is where you can find information, the public health sites that actually provide you where outbreaks are happening and what the various health agencies are actually watching as potential outbreaks. I mentioned that again when we talked a bit about H1N5. So remember, viruses are not self-sustaining. They reproduce within a susceptible host. So they're considered an obligate intercellular parasite. Why? Because they can't survive without that host. So it's obligate. They have to get into that host, so they're intercellular, and they basically use all of the building blocks and all of the protein synthesis, et cetera, and all the various metabolic pathways of the host to be able to make multiple copies of themselves. And they cannot synthesize constituent molecules like nucleic acid or proteins outside the host. They are infectious. They're transmissible between hosts, but their transmission between hosts is very specific. And their entry into particular cells is very specific and is based on what surface molecules that virus has on either its capsid or its envelope, and what cell types have the appropriate receptor for those particular surface molecules. They can utilize the machinery of the susceptible host to be able to replicate, make the proteins they need so that they can actually build multiple copies of a virus. And really, they represent a very diverse group of agents. So this just shows you some of the general sizes to give you an idea. So this is part of a red blood cell. This is E. coli. And you can see that viruses come in various sizes. Here you see a bacteriophage that infects bacteria. Here's another bacteriophage. This is the size of a prion. This is a virus that infects the tobacco plant. And you can see that they come in various sizes and in various forms. So the smallest viruses are about the size of a ribosome. The largest, which are generally the pox viruses, are actually almost the size of a small bacterium. So they do range in size substantially. And we can use several criteria to categorize viruses. So one of the primary ways we categorize viruses is by their size and morphology. So we consider, is there an envelope or is there not an envelope? So we can have naked viruses that simply have the capsid. They have no lipid envelope. Or we can have an envelope surrounding the capsid that contains the nucleic acid. So all viruses are going to have a capsid in which the nucleic material is inside the capsid. Not all viruses have an envelope. The envelope, as we will discuss, is composed of lipid and actually comes from the membranes of the cell they infected. The surface proteins that are placed in the envelope are actually made and encoded, are actually sort of encoded by the virus and placed into that envelope. Okay. So the virus needs to be able to make its capsid, which is viral encoded. It needs to make its surface molecules, and it may need to make specific accessory proteins to carry out genetic replication. So if it's not a straight DNA virus that's double-stranded, where it can just use the machinery of the host, it often has to bring an accessory factor to help carry out the replication of the genome. The fact that some viruses are naked versus enveloped is important. Enveloped viruses that have lipids are easier to kill. Simple hand washing does the trick. Naked viruses also can be killed by detergent, but they're harder to get to actually break open because the capsid is proteinaceous, it's not lipid-based. And so it's harder to actually, for most of those viruses, to have a easy removal. Those viruses, whether they're a capsid, so here the spikes are not being shown, but if it's a naked virus, there will be spikes on it. If it's an envelope virus, the spikes are going to be in the envelope. It's the spikes that allow it to attach to the specific host to be able to infect that host. Okay? So the first place we start, is it a enveloped or is it a naked virus? Then we can very easily classify the shape of the capsid. So in this case, this and this are classic icosahedral capsids. But you can also have spherical capsids, helical capsids, or what we might call complex capsids where they actually have different components. So the bacteriophage is very much a complex capsid because it has a place where the genetic material is. It has a tail and spikes that allow it to actually be able to land on bacterial cells and actually infect it. I'll remind you again that the lipid in this membrane comes from the host membranes. Nuclear, plasma membrane, endoplasmic reticulum, depends on where it's budding from. That will actually generate the envelope. So this just shows you the various shapes. So here we have a helical. So you can see that the genome here is RNA. It's wound, and surrounding this wound RNA is a helical capsid. This is what it looks like in EM. This is tobacco mosaic virus. Here you see a classic virus that's icosahedral and doesn't have, by the way, although we're not showing the envelope here on this diagram, and this diagram tells us what? These are naked viruses. This is adenovirus, classic icosahedral DNA-based virus. Common virus that infects us, gives us characteristics that are common to what we would define as common cold, although really the rhinovirus is the virus that is specifically identified as causing a common cold, but the problem is many viruses give the same symptoms. Any virus that affects your respiratory system, especially your upper respiratory system, is going to give similar symptoms. Then we have here the influenza virus. This is showing the envelope with the spikes, and then we have a helical capsid, and in this case, the genetic material inside that helical capsid is actually in segments. So it's not one piece of RNA, it's actually in segments. And this is what makes the influenza virus highly problematic, because one influenza virus can come together with another influenza virus that have slightly different segments, and they combine and they get a totally reassortment of the actual segments, and now you have a virus that is perhaps brand new, or one that hasn't been seen in a long time. And here you have a classic bacteriophage. This is actually T4. Lambda looks similar. You have a icosahedral capsid with the DNA, double-stranded DNA in the capsid, but you also have this very complex structure with a tail, with tail fibers and spikes, because this is going to literally land on the bacteria cell like a lunar landing and actually shrink its spikes and then drill a little hole in the cell wall so that the DNA can actually move into the prokaryotic cell. So some viruses actually enter into the host cell. Some viruses, only the DNA or RNA enter the cell. It depends on the particular virus as to the mechanism of entry of the genetic material. So here you see a classic icosahedral capsid. So this has very specific geometry, okay? And you end up with a scenario where capsids are made up of the same protein. So it's not made up of different protein. It's one single protein, but can be arranged in different ways, from icosahedral to the other kinds, the other structures I just showed you. Again, we can have naked icosahedral viruses, or we can have enveloped icosahedral viruses, for example. Here again, we see the tobacco mosaic virus in a little more detail. So what you're seeing here is this is the genetic material. This is the actual helical structure, and you can see that how the components of the capsid, the capsomeres, actually surround the genetic material to make that helical structure. Here's our bacteriophage again, showing us the structure. So that means this genetic material has to get from the head through the tail to the base plate. So here you can see a number of bacteriophages attaching to the cell wall of a bacterial cell. And if you look really carefully at this diagram, you can see that the tails are different lengths because it's starting to collapse on itself. And if you look really carefully, you can find a couple of heads that have already injected their DNA into the host, like, for example, this capsid right here. So actually it lands, the tail shrinks, the tail spikes hit, the genetic material gets actually injected into the host. So again, what makes it complex? It has an icosahedral head, a tail. It has no envelope. Here we have an example of, this is actually SARS-CoV-2. And this is it. So this is a sort of a colorimetric version of it, and this is actually observed under the electron microscope. So you can see the helical genome and the capsid surrounding it. So this is an RNA-based genome. You'll note something important, because a lot of people don't quite make a mistake when they start talking about mutations in COVID-19. Is this a segmented genome? So it can't do what the influenza virus does. Yes, it undergoes mutations, and those mutations affect some of the characteristics of the infection, but it is incapable of going through a complete resortment like the influenza virus is, which actually at least is an advantage, because that adds a whole other layer of problems for viral infections. You can see that there are some important proteins in the envelope. There's a protein called the M protein that actually is seen with a lot of viruses that helps support the envelope. And then you see some other proteins that are important, but the important protein that allows it to attach to the surface of a host cell is actually the spike protein. And those spike proteins are universally glycosylated. So they can attach to important proteins in the extracellular matrix, especially of eukaryotic cells, where you find a lot of glycoproteins, or other mammals. But the point is, it is the spikes that determine the specificity of that virus. So that spike determines where COVID-19, the SARS-CoV-2 virus can attach, predominantly to in your respiratory system. But there's one other place it can attach. Does anybody know? Sorry? What are one of the other strains? So when you were listening to all these symptoms, they were all respiratory: cough, headache, fever. But there was another one that seemed a real outlier. No, fatigue would fall under that category. Diarrhea, because there's also a receptor in your gastrointestinal system. Not in as high a level as you find in the respiratory system, but it does exist. So where the symptoms occur tells you where the virus is actually attached, what cell types the virus is attaching to. This is the influenza virus, again, where you can see the spikes. You can see the segmented RNA genome and each segment surrounded by the helical capsid. And what you're going to learn about, if you don't already know, is influenza has 2 different spikes on it. And those spikes become really important for where influenza can actually attach. And we'll talk about that in a little more detail next week. So again, I've shown you a few different viruses to make a point that while classically we think of a spherical but more commonly icosahedral or helical, there are viruses that don't easily fit into these descriptions. Classic example, the bacteriophage, but another important one is the pox viruses, which have a very pleiotropic capsid, so they change shape. So there are some variations to the kinds of capsids that can actually form. So, is it enveloped or is it not? What is the shape of the capsid? The next one is, what is its host range? Okay, so what kind of cells or organisms does this virus infect? Remember, a virus must bind or adsorb to infect and grow to reproduce. So bacterial viruses are often given the name bacteriophages, and those that infect E. coli are often called coliphages. So a bacteriophage has the most common one has that complex structure, but there are some variations amongst the bacteriophages. A plant virus generally almost always has a specific range. So the tobacco mosaic virus only infects tobacco. Okay. Animal viruses also have specific ranges. So we have human viruses. We have avian viruses. We have viruses that only affect your cat or your dog. And those that are very specific to very specific lineages cannot infect you. So when you say, I gave my particular cold to your dog, it is highly unlikely. There are some viruses that will infect both of you, but generally speaking, many of the viruses that infect you will not actually infect the dog. Dogs and cats could not be directly infected by the COVID-19 virus. With enough virus, you could make them a little bit sick, but they couldn't pass it to you. Okay, so this is an important concept. Viruses attach very specifically to usually very narrow host ranges. There are some viruses that have wider host ranges, but most of them have a narrow host range. Also, yep?
What about like zoonotic spread?
The zoonotics, there are viruses that can spread to different organisms, yes. There are subpopulations of viruses that can move from different organism to different organism, yes. But for example, some of those viruses may have to mutate in the process. So for example, let's take bird flu, for example. It's an influenza virus. It infects birds. You as a human, if you come in contact with enough sick birds, may actually have some symptoms. But you, at the present time, have no ability to spread that to another human. So there's usually some kind of, very often, not always, some of them can do multiple organisms, but sometimes there's a mutation that takes place. So a pig influenza gets together with a bird influenza and a human, and they resort, and you end up with a human, and there's also a human influenza in there, and you get a different virus. That can't always happen, but that specific example is unique to influenza, right? Whether or not the coronavirus mutated to infect humans or not, I don't I honestly don't know whether we know because we don't know where it came from. Okay? We still don't know where it came from. You can pick your favorite theory or your favorite espionage theory over where it came from. I do not think it was a legitimate release from a lab, but anyway.
So building on that question, I was gonna ask, could there ever be like specifically for the flu because of how it like recombinate? Is there any way, any like composition of like the hemoglobin and neuraminidase that could like affect humans and animals, like the same combination? Or would it just be specific and then
The answer probably is with the right recombination of the surface, that probably it could actually infect both organisms, yes, yeah. Without all the specifics, because some of the human influenza viruses really were bird and pig, and the recombination, the human and bird recombination event occurred in a pig, with the pig virus there too. So they were also sick. So without knowing the specifics, my answer would be probably yes. There's a lot of things we don't understand about viruses. That's what makes this very hard to actually predict what really is going to happen. Okay, so shape of the capsid, naked, enveloped. We also have the type of nucleic acid. Is it DNA-based? Is it RNA-based? Is it double-stranded? Is it single-stranded? Because all combinations are there. It can be RNA, DNA, linear or circular, double-stranded or single-stranded, contiguous or in segments. So one piece of DNA or 4 pieces of DNA, or one piece of RNA or 5 or 6 segments of RNA. So you have various variations. So just as an example, here are a variety of viruses that infect bacteria. You have a small number that are RNA-based, that one is single-stranded, one is double-stranded. We have several that fall within the single-stranded DNA, but a large number of them fall within the double-stranded DNA because, of course, the double-stranded DNA viruses are the ones that can utilize the host very readily without having to have accessory proteins to help with the process. All right. So we actually can get even more confusing with our viruses. So yes, it can be DNA, single or double, RNA, single or double, circular or linear segments. But we also have viruses that are RNA viruses that go through a DNA intermediate, for example. So we have the single-stranded RNA viruses, which are your retroviruses that enter the cell as a single-stranded RNA. They are reverse transcribed into ultimately a double-stranded DNA molecule that enters the nucleus, gets replicated, leaves, and then is packaged as an RNA molecule again. We also have the hepatinoviruses, so you can guess which location this virus infects, a liver, but we have a collection of viruses that infect the liver, and they're not all double-stranded DNA viruses. So this is where naming viruses can be problematic. You call this a hepatinovirus, and you think this is the only virus that infects the liver, but it isn't, as I will explain to you. It is one that will go through, it's a double-stranded DNA virus that can go through an RNA intermediate. So there are variations to the way in which these genomes can be processed. And if we start to look at and this is just a small number, obviously. So here we have a variety of different viruses showing you both animal and bacteria and plants. And we can see that, animals, if we just concentrate on the DNA, single-stranded, double-stranded, but we start to see that if we really look at DNA viruses and we put a big chart up and ask what is the most common form of a DNA virus, it's going to be double-stranded. Okay? But there are variations. When we look at RNA-based viruses, yes, we can have double-stranded. Yes, we can have single-stranded. But when we start to actually look at the populations, we'll see a tendency towards what? Single-stranded or double-stranded? what do our cells use? Single-stranded or double-stranded? Double-stranded RNA? Single-stranded. So you'll see a trend towards single-stranded. Especially amongst the animals, the interesting thing is amongst animals, you'll see more RNA-based viruses than DNA-based viruses, which is very interesting as well. It also shows that they can be of very different sizes. So you have the genome sizes. It's not, we're going to concentrate on only a couple of these as we go. This is just to give you a vision of the kinds of organization you can see. So here you see that the number of molecules that can be present. This is a structure, this is a base pair, and the number of molecules that can be present. So you can see in the Rio virus, it has 10 different segments, for example. The cowpea mosaic virus has 2 different segments. And then I've already shown you the influenza virus that also has different segments. So lots of different variation amongst the viruses. Then we have the way in which they carry out their synthesis. So amongst the viruses that consist of a single-stranded RNA, they can be divided into 2 groups, a sense RNA virus and an antisense RNA virus. Okay, so positive, plus, or sense, that means the sequence of RNA packaged into that virus. So as that virus entered that cell, it can be directly translated because it's a plus strand. It will have to go through an interesting combination of modification, making a second strand. So the plus strand would have to produce a minus strand to actually make a more plus strand for actual replication. But it means that virus can immediately start synthesizing protein. It can be directly translated. Examples of viruses that fall within this group are the enteroviruses, which are polio, the ruby viruses, which is rubella, which sometimes have been given the name German measles, rhinoviruses, which was originally identified as the common cold virus, and rhinoviruses have so many different subtypes that you can't make a vaccine to it, but you generally do not get the same rhinovirus in the same year. So you have protection against it for a year. So if you get another rhinovirus, it's a different subtype. But there are many viruses that have the same characteristics of the common cold. And why is rubella such an important thing to have been vaccinated against or know if you've actually had it? Because it's a relatively mild viral infection for those that have it, except for a specific group of people, and that's pregnant women, because it can cause severe damage to the fetus. This becomes very important for people who are going to be pregnant, want to be pregnant, or are pregnant to be aware. Are they vaccinated? Is there people in their sphere that have not been vaccinated? Because it can cause severe problems with the fetus. The negative, the minus, or the antisense, that sequence that's packaged is complementary to the viral RNA, so mRNA, so that it can't be directly translated. You have to change, it has to be made into a plus strand, and then that plus strand would be used to make more minus strand for making more genome, and then it would carry out protein synthesis. Examples of viruses that are, again, human-related: Lassa virus, which is rabies; paramyxovirus, which is mumps; the orthomyxoviridae, which is the influenza virus; or the morbillivirus, which is measles. So that's just demonstrated here, just to give you a variation of the ways in which synthesis can occur. So here's your classic double-stranded DNA. There are 2 different classes. They move directly into mRNA. Single-stranded DNA viruses of class 2 obviously need a double-stranded DNA intermediate because our cells don't know what to do with the single-stranded DNA sequence. So we have the class, by the way, of this class 7 is actually got a transcription followed by a reverse transcription. Don't want to get into that much detail except to say replication and transcription in viruses is highly complex. It's not simple the way we've learned in eukaryotic cells in terms of, DNA, double-stranded DNA goes into messenger RNA that's then used to make proteins. You can see that the RNA viruses, double-stranded, plus the plus versus the minus are different classes, and they can generate the mRNA in different ways. And then we have the single-stranded RNA viruses that are in the retrovirus category, which require a reverse transcription. So again, lots of variation in how these viruses do what they do. Okay, remember this is an overview, so the details to which I'll go through a couple more viruses next week just to visualize some of this, but it's to give you a vision of the complexity of what you have to address when you deal with viruses. Another category or criteria you can use is the disease they cause, or the syndrome. This is a little bit more problematic. Sometimes it's fine calling it a human immunodeficiency virus, HIV, which causes AIDS, the influenza virus, which causes the flu, or the SARS virus. But many of these names don't work very well because there are viruses with broad host ranges that may not necessarily help you define the host. And for example, you can talk about an E. coli virus, but there's approximately 10 different coliphages that can actually infect E. coli. And most importantly, many viruses can produce the same symptoms. So for example, rhinovirus, which is the common cold virus, as well as several other viruses, can cause the same symptoms. So you say you have a cold because you have a set of symptoms. It doesn't mean you have a rhinovirus. You could have an adenovirus. You could have a coronavirus, because we get virus infections on a regular basis too. So the most informative and least ambiguous is genome type, mRNA synthesis type or pathway, and the morphology. So based on what we just talked about, the criteria used to group viruses include the shape of the nucleocapsid. Yes or no? Yes. The presence or absence of an envelope. Yes. The type of nucleic acid that serves as the genome. Yes. Okay, so all of the above are correct. So I'm going to end with this, and I'll just pick this up next week. Little aside, the longer you lecture in years, the less material you cover in 2 hours. It's like counterintuitive. You'd think you'd be able to do more, but you end up doing less. Anyways, that's just an interesting aside. Talk to anybody who's lectured for a very long time. They'll say, I just don't cover as much material as I used to in 2 hours. Anyway, so this is just to help you where you can look at characteristics of a virus and remind you of the characteristics of a cell. It just will help you when you're summarizing your notes. So I'll pick it up with this table, which demonstrates this idea of role of RNA viruses in mammals next week. Send me your name and email. You realize that you're under no obligation to do anything. Taking conflict courses does not absolve you of attending the project. I know, but you're asking us to accommodate you because you took a conflict course. That's my point. That's a choice you make, and if there's in-class exams and you're supposed to be in 2 places, then you lose your grade. There's very few faculty that will actually say yes to that, because you're not supposed to take conflict. That's a choice you make. As soon as you saw there was a conflict, you should change your lab if possible, or decided on which course. Because we're not responsible for actually accommodating you for that. Okay? So, send me the email again with your date, and I'll look at the Tuesday class, but it would have to be the morning. But understand, I don't have to do this. And there are, if you were to go to DPES and ask them to do this for you, or psychology, they would not do it for you. Yeah, you're not supposed to take conflict courses, okay? All right, so send me the email again. Yep. Say what? Do you see an announcement that there was any homework? Then no, you don't. Okay? You will know when you have to do something because there'll be an announcement, there'll be an assignment there. Okay? All right. Question? Well, what are you seeing?
So, I think I have an idea. I think I know what my next is, and I know that it's gram positive, right? So I know it's supposed to be.
You know, why do you know it's supposed to be?
Well, because here's the thing. Okay, so the color of it kind of indicates the
You mean the color of the culture?
of the culture.
Okay, well, that's an observation.
Yeah. And
So, what organism do you think it is?
ambrosius.
Okay, so then what do you see in the gram stick?
So that's what I'm asking because it came out like pinkish. It had purple parts and blue
It's
parts.
because you're getting variable staining, which likely means you're not applying to the entire slide. And when you decolorize, and if you have too many cells, and oftentimes with Micrococcus you end up with stacks of cells,
Stacks of cells.
you end up not staining all the cells.
Not staining all the cells properly.
Fewer cells gives you better staining.
So just a lighter, like when I'm grabbing this.
Yeah,
blue,
take less
lighter,
cells.
Less cells.
Yes.
But
And then, when you do decolorize, and this is just a general rule, what I see is students like just sort of putting it on, but not, when you add the 95%, they should sit on your slide rack, and you should see the 95% falling off the slide,
like falling off, right?
like it's a little waterfall.
Would it be good to like maybe move the slide up a
No,
little bit?
because then you don't hit the
You
whole
don't hit the
slide.
whole slide. So you want to leave it flat.
Yeah, and then just keep dropping it,
keep dropping
and watch
for
the,
5 seconds. And then
keep going.
with the water. And when
Not
rinse-
water.
Oh, sorry.
The
No, but
alcohol.
afterwards you have to rinse it, right? In
Yes,
between each.
but the key is if the decolorizer does not hit all of the slide and you've got a gram negative, then you're gonna get variability too. Yes.
Okay, and when rinsing, you put it maybe at the top edge you would recommend and not directly on the actual smear, like just kind of let it trickle
Oh,
down.
yes, you can just rinse that way.
Yeah, but not for too long, right?
No, you're just rinsing off. Theoretically, there really shouldn't be any
Shouldn't be
stain.
an issue.
That's after the saffron.
That's after the stuff. Oh no, are we rinsing off each part, like Crystal Violet,
You can, but that shouldn't be very long at all.
Shouldn't be long at all?
You don't even have to